In our last lecture, we discussed the seedless plants. In this lecture, we're going to talk about the seed plants. These are also called our gymnosperms and our angiosperms. So taking a look back at our chart that shows the major plant divisions here, as I just said, we discussed last time the seedless plants. Now we're talking about the seed plants. These are called our spermatocytes. Sperm means seed, and we have two different types, the gymnosperms, our naked seed plants, gymno means naked, and then the angiosperms, our vessel seed plants, angio meaning vessel. Where these all fall out phylogenetically is quite interesting. So last time we talked about the non-vascular plants. This is the clade represented by the colors red, green, and purple. That is our non-vascular plant clade here. We have our bryophytes, like our mosses, as well as our lycophytes, most of which are extinct. But now we're getting into a distinct part of the vascular plant clade. So last time in our lecture, we also discussed the horsetails, which are vascular plants, and a sister group to that specific type of vascular plant are our spermatophytes, our gymnosperms and our angiosperms. So this is just where all of this fits in genetically when we're taking a look at phylogenetic relationships. When the first seed plants emerged about 400 million years ago, it was in the Devonian period. And it was not until about 270 million years ago that we got the emergence of our gymnosperms. The emergence of the gymnosperms occurred during the late Jurassic. This was during a time in Earth's history when we went from predominantly wet and moist climates to one that was a bit drier in the Mesozoic. So we had an environmental shift to a drier climate. This acted as an agent of natural selection to select for plants that did not necessarily rely heavily on water for reproduction and growth. And therefore, we have the widespread emergence and radiation of our gymnosperms. So this again began about 270 million years ago. The first group to pop on the scene were our Ginkgo galleys group. The ginkgo is represented today by only one living representative, and that is the Ginkgo biloba plant. This may be a familiar name to you because it is used in some therapeutic agents and holistic medicine shops, but Ginkgo biloba is the one and only living representative of the first gymnosperm to hit the scene in the Jurassic. We also have represented in this group plants like the cycads, which sort of look like a palm tree to me, but it's not. So this is where we saw the emergence of gymnosperm diversity in the Jurassic. Moving on to the angiosperms, these are our flowering plants. We also know that they are a direct sister taxon to our gymnosperms here, both of which are our spermatophytes. We know from fossil evidence by the mid-Cretaceous, a staggering number of diverse flowering plants crowd the fossil record. What's interesting is that during the same geological period as when we see the radiation of our flowering plants, we also have the appearance of many modern groups of insects, suggesting that pollinating insects played a key role in the evolution, the dispersal, and the radiation of our flowering plants. And what I mean by radiation is we have a massive amount of different forms. There are about 300,000 different species of angiosperm. So that's a lot of diversity, and it's actually only second to the number of different insects we have on the planet. So therein lies some hints between the relationship between insects and flowering plants. We have fossil evidence of some of the first representatives of our flowering plants, mainly as imprints where soft sediment gently caresses the creases and folds of a fossil, or what will be a fossil leaf that gets preserved for us to find later. So we have flowers that come in a bewildering array of shapes, sizes, colors, smells, and arrangements. And most of these flowers have what we call a mutualistic pollinator. So a specific type of pollinator that is only attracted to a specific type of flower, therein lying this mutualistic relationship between the two. So what contributed the most to the rise of seed plants? If you remember back to our discussion on bryophytes, our mosses and our ferns, those organisms are highly reliant upon water for reproduction and dispersal. But the presence of seeds and pollen within our seed plants allowed these plants to be freed from their dependence upon water, thereby allowing them to conquer dry land. And so the presence of seeds and pollen eliminated the dependence upon water for reproduction. And unlike our bryophytes and our mosses, where we have the dominant gametophyte generation, in our seed plants, the dominant generation is the sporophyte generation. And here we have on the right just the massive amounts of diverse modalities by which our seed plants are able to disperse their seeds in their environment. So moving now and focusing in on the gymnosperms here, we have 4 major divisions. Number 1, we have the Cnidophyta. Only 3 families of the Cnidophyta are alive today. This is one of them here. In addition, we have the Coniferophyta, or the conifers. Everybody should know what a conifer looks like. This is our pines, our spruces, our firs. We also have the Ginkgophyta, most notable the Ginkgo biloba, our only living representative within this group. And then our Cycadophyta, our cycads, which again is imaged here on the bottom right. So I said before that the term gymnosperm means naked seed. And this just means that the seeds are not housed within an ovary, but instead they're housed within specialized modified leaves called sporophylls. Okay, we all know what a pine cone looks like. So this is the appearance of a mature female pine cone. The projections moving out on the left, right, and top are called sporophylls. These are modified leaves, believe it or not. And then within here, we have what is called the sporangium. The sporangium is where our spores are housed. So of course, we have to talk about the life cycle of the gymnosperm. Like our bryophytes from the last lecture, we also have an alternation of generations within the gymnosperm. Again, what we're able to recognize with our eyeball is the sporophyte generation, which is a diploid organism. We call gymnosperms heterosporous. Okay, hetero means different, and sporous refers to spores. So what this means is that a gymnosperm contains 2 types of spores. What could those 2 types of spores be? It's the male spore and the female spore. The male spores are called microspores because they are microscopic. They're very tiny. By contrast, our female spores are called megaspores in the gymnosperms. And that is because they are pretty big. We have ovulate cones, which are the female cones. They grow in the upper branches of the sporophyte organism, where they can be fertilized by pollen that is windblown. And then we also have staminate cones. And these tend to be lower on the branches here, and they are our male cones. So this is a mature tree that we would call monoecious. What does that mean? Mono means one, ecious means home. So a monoecious plant means one home for both types of spores. Okay, so we would call this individual bisexual because it produces both female cones, the ovulate cones, and our staminate cones. Okay, it produces both cones with pollen and with egg. So this is a monoecious gymnosperm. It contains both within one plant. By contrast, you can be a dioecious plant. Well, if monoecious means one home, dioecious means two homes with that prefix di, meaning two. Okay, so they are unisexual individuals. One individual will produce only pollen, and the other individual will produce only eggs. So they are separated individual by individual. Okay, so that is the difference between monoecious plants and dioecious plants. We also have this exhibited in the animal kingdom as well, and we'll get to talk about that in future lectures. Now let's talk about angiosperm diversity. Angiosperms are classified into a single phylum called the Anthophyta. Modern angiosperms appear to be a monophyletic group, meaning there's an ancestor and all of its descendants when we're looking at a phylogenetic tree, such as this one here. Within the angiosperms, there are three major groups. Number one, we have the basal angiosperms. These are represented by the magnoliids, the most common being the Magnolia grandiflora. And so when we take a look at the cladogram representing the Anthophyta, the magnoliids fall over here. We also have the monocots and our eudicots, or just dicots. And the distinction here has to do with the type of seeds and morphology of their roots and leaves and vascular bundles. So in the next couple slides, we'll talk about the difference between our eudicots or our dicots and our monocots. As a zygote grows into the embryo after we have a fertilization event, the first leaves that form are called the cotyledons. Okay, this is another name for seed leaves. So when we're taking a look at the differences between monocots and dicots in terms of their seed leaves or cotyledons, monocots have one and dicots have two. Okay, so monocot and dicot is actually shorthand for monocotyledon and dicotyledon. And cotyledon, again, being that seed leaf. We have different examples of monocots and dicots represented here in this figure that I pulled from your textbook. So monocots are represented by corn and lily, whereas our dicots that have two cotyledons, they're represented by beans and oaks. We also have differences in relation to leaf and root patterns. So here on the left, the leaf venation pattern in a monocot is parallel. Okay, so this looks something like a grass, for example. By contrast, our dicot has more of a net pattern that looks more veiny. Taking a look at the root patterns that distinguish our monocots from our dicots, the monocot is more fibrous with lots of little tendrils poking out from the organism where it first enters the subsurface. And then our dicot has this huge taproot, okay? That is not something we see present in the monocot. The monocot is more fibrous. The dicot has this very thick taproot. Moving now to flower parts. Here on the left, the monocot comes in groups of 3, represented here. And then the dicot comes in groups of 4 or 5. The vascular bundle position is also important in distinguishing monocots from dicots. The monocot has the vascular bundles that are just sprinkled scattershot, whereas in the dicot, it is arranged in a circle. Finally, taking a look at stem types, the monocot tends to be herbaceous with its stem, while the dicot tends to have a woody stem. And it could also be herbaceous as well, with lots of little leaves protruding from the main stem. But it has the option of being either herbaceous or woody. with that woody stalk leading up to the different branches here. So here's a table that I lifted from your textbook that gives you a summary of monocots versus dicots. There are also other images that should hopefully help you. Moving now to flowers. Flowers, believe it or not, are also modified leaves. Okay, so they have a specialized purpose, usually to attract pollinators and help in reproduction. So although these flowers tend to vary greatly in their appearance, virtually all flowers contain the same structures. We have sepals here, we have petals, we have carpels or the gynecium, and we have the stamens or the androecium. Okay, a whorl of sepals underneath our petals collectively is called the calyx, and that is located at the base of the pedicle and encloses the unopened bud that you probably see growing on your rose bush, for example. If you're tracking your roses, the first whorl of sepals that you see, that is called the calyx. Sepals are usually photosynthetic organs, although there are some exceptions. We have the collection of petals. Collectively, the collection of petals is called the corolla, and this is located inside the whorl of sepals, and of course they can display a wide variety of colors to attract their pollinators. The sepals and petals together form what we call the perianth. Okay, so the corolla, again composed of petals, and the calyx, again composed of sepals. Now moving on to the sexual organs, we have the carpel that produces the eggs or the ovule, also called the megaspores, inside of the ovary. These ovules will ultimately develop into a seed. We also have on the right here the stamen, which produces our microspores by meiosis, and they develop into haploid pollen grains or male gametophytes. We have a TED Ed video that describes the fertilization process of angiosperms, and I recommend that you go through it. Sorry, I couldn't take an actual screenshot of the TED Ed. For some reason, the website is down right now. Now let's talk about reproduction in our flowering plants. We know that the adult phase, the sporophyte phase, is the main phase of the angiosperm life cycle. And like our gymnosperms, the angiosperms are also heterosporous, meaning that they have two types of gametes. We have the microspores and the megaspores. The microspores form inside the anther, and the megaspores form inside of the ovary. So inside the anther's microsporangia, we have the male spores that are formed by meiosis. This is generating haploid spores. Okay? These haploid spores undergo mitosis to create what we know as pollen grains. Each pollen grain has two types of cell. It has the generative cell that's going to become the sperm, and the tube cell that's going to become the pollen tube through which the sperm travel. So once we have the joining of the pollen to the sticky stigma, we have this weird phenomenon where there are chemical signals released by the ovule to say, hey, come on sperm, I'm over here. So what happens is this tube cell begins to form a pollen tube. This pollen tube grows eventually through this long shaft of the ovary, finally reaching, let's zoom in over here, finally reaching the ovules and the other cells that are here. Okay, so this tube, once established, allows a direct route through which the sperm can travel. These sperm are going to travel to fertilize the egg and also join with the nucleus of the cell within the ovule. And this is what is known as a double fertilization event. and there are videos for you to watch to help make this a little bit more clear than what I've been able to provide to you with the schematic alone. So moving on to fruits. So as the seed develops, the walls of the ovary begin to thicken. Okay, so fruits are actually ripened ovaries. They're really, really big ovaries. And why are these fruits so important? They're important agents of seed dispersal. So you probably know that mammals are really good seed dispersers. Our tummies can't digest seeds, and neither can the tummies of monkeys, for example. So animals that eat native fruits, they do not digest the seed. Instead, that seed gets passed through their digestive tract and excreted to fertilize the ground wherever they are. So there are massive different types of fruits, and a lot of the things that we consider vegetables are actually fruits. Things like zucchini and eggplant and bell pepper, tomato, those are all fruits because they are ripened ovaries. The winged maple helicopter seeds that you see in the fall, some people call them whirligigs, those are also fruits. So a vast amount of fruit diversity out there. And if someone gets into an argument with you about a tomato being a fruit or a vegetable, you know that it's actually a fruit.